Hello everyone, today we'll talk about lecture 2, similar amount and type of DNA in different cell types. This lecture is lecture number 2 in the course Molecular Genetics 1. It's Molecular Genetics MLGN 301 in 2016. This lecture we will talk about many issues. All the information that of this lecture have been extracted from the book Gene Regulation, 5th edition, David Lec2. And in this lecture we will learn how DNA of different cell types is similar in both amount and type. And the outcome of this lecture that we should understand by the end of this lecture the DNA of different tissues and cell types is generally the same in specific organism cases. Also we'll understand cases of DNA loss and understand that the cases of DNA loss do occur in specific cell types in particular organism, and these are exceptions, of course, caused by special requirement in particular situation. And we will see that gene regulation in the end of this lecture operate to produce different amount of different RNA in different cell types from the same DNA. In this case, we will learn in the end of this lecture, we will learn the differences between four major techniques in this regard. We will learn the difference between Western blotting, Southern blotting, Northern blotting, and dot blot. In this lecture, we will see that these are the topics which we will discuss. That the DNA loss, is a mechanism of gene regulation, and we could study that on three different levels. The first level is chromosomal studies, the second level is functional studies, and the third level is molecular studies. As we see that the DNA loss are a mechanism of gene regulation, and the scientists have proposed a model for gene regulation by specific degradation of genes whose products are not required in this particular tissue. So they put a model, and they want to know if this model is a real model or not, if this working or not. So they put that model, that gene A and gene B, this in a specific tissue, tissue number 1, and we'll see that if we don't need gene A in this specific tissue, this gene will be degraded. But if we need gene B will stay and only degraded if we don't need it in that tissue. At the same time, we'll see that in tissue 2 we need gene A but don't want gene B, so simply we just discard or degraded the gene B will be degraded. So we only use the genes that are needed in that specific tissue. So that model was proposed by some scientists that is the way of gene regulation. But of course we saw after that is not the right one, but it only works in very special cases. But in most cases and most tissues, genes are not degraded by that mechanism. So let's see how things are working in most cells. As we see that a variety of evidence suggests that selective DNA loss is not a general mechanism, as we just said. Such evidence is available from chromosomal, functional, and molecular studies, which will be considered in turn. Let's see the studies, the chromosomal studies. In general, we see that the chromosomes in most of the cells from different cell types of the same species, we will see is indistinguishable numbers and sizes and banding pattern of chromosomes. So we can see there is no we cannot distinguish between different cell types from the same species from the chromosomes so the sizes can be as the same, numbers are the same, banding pattern of the chromosomes are the same in all the tissue from different cell types in the same species and the same organism. So So that is clear that the chromosomal study indicate no differences. So you will see that the pattern obtained from, for example, liver cell or a brain cell are very similar, and the people cannot distinguish between them by cytogenetics techniques. Karyotype, which we see right here, karyotype of the chromosomes on both cells are the same, so it's provide actually strong evidence against the loss of the whole chromosomes of specific parts or specific parts of chromosome visualized by G gamma bands in particular differentiated cell types. In this case, this chromosomal study actually argue against selective DNA loss and a general mechanism of gene regulation. So chromosomal study in general, as we see, it's provide a clear evidence that most cells have the same amount of chromosomes, have the same amount of DNA, and there is no such evidence from chromosomal studies that there is a DNA loss involved in the regulation of genes in different cells. So in this case, nevertheless we found that there is a small number of cases such as such a loss of DNA have been observed in particular cell types, and these will be considered as we see now. These are exceptions, as we said. This example number one, loss of nucleus in mammalian red cell blood cells, and this is the best known case occur in the development of mammalian red blood cell, which is highly specialized cell containing a large amount of blood pigment, hemoglobin. In this, we know that the function of these cells are to transport of oxygen in the blood. So in this case, we saw this special case that the cell, simply red blood cell, get rid of the nucleus. So we and have this new cell without any nucleus. So we call it enucleated erythrocyte. This enucleated erythrocyte is actually a cell without a nucleus, and we can see that we can found highly active RNA which produce the hemoglobin gene. So this extrusion of nucleus from erythroplast resulting in enucleate erythrocyte. So as we see, hence the tissue-specific pattern of protein synthesis observed in this kind of cells is achieved not by selective gene loss, but by the process which earlier in development resulted in high level of transcription of the globin genes and production of long-lived stable messenger RNA molecule. As we said, we have here a long-lived high level transcription of globin gene. It's a messenger RNA. This messenger RNA gives production of long-lived stable messenger RNA. It exists here inside the new cell, but there is no nucleus. So this extrusion of that nucleus, of course, only in this case. We cannot find it in other cell or other cell types in other tissues. So in this case, we can see that because the cell doesn't need the other genes, so it simply gets rid of them. Here in example 2, we'll see loss of specific chromosomes in somatic cells. However, a number of such cases in which specific chromosomes are lost during development have been described in other eukaryotic, including some nematodes, crustaceans, and insects. In these cases, actually, we see not the whole nucleus will be lost, but actually some chromosomes. only some chromosomes. So the other cells will have the rest of the chromosomes. So let's see these cases. For example, in the early development, of course, of embryo and in the cleavage stage, the somatic cells lose certain chromosomes. So, which only the germ cell which give rise to subsequent generation retaining the entire genome. So simply we'll see that the germ cell contain all the chromosomes, the total, the full number of the chromosomes in the germ cells, but in other somatic cells we will see different number of chromosomes due to chromosome loss that happen in this early developmental stage. See that example, for example, this in nematodes, we can see that the zygote at that time, this zygote is simply, when it divide, it's a normal division. They make unequal division. Some chromosomes are lost, give rise to somatic cells. So the somatic cells have a less number of chromosomes. So the number of chromosomes in somatic cells in nematodes, for example, is less than the original number which retained in the germline. So the germline, the germ cells, contain the actual number of the chromosomes in the nematode, but the somatic cells contain less number of chromosomes. If you just take the zygote and try to do a centrifugation and to have a cell divides in wrong place, for example, you find there is no chromosome loss from either cells. This is a normal, for example, in the cell culture or whatever. You can see that this number will not change, but in the process in speciation of differentiation of a specific cell, for example, from germ cell to another specific cell, we can notice this different number of chromosomes in each type of tissues. So although cases of DNA loss observable by examination, as we saw now in the last two cases, some in some organisms, of course, but the majority and the vast majority of cases, no such loss of DNA is observed. So the two exceptional cases that we talked about, the nematodes and the other one, these are actually for example, in the red blood cell or in nematodes, these are actually exceptions. And in the most tissues and most organisms, we cannot find such loss in their tissues, as we will see. Okay, the number two in this lecture is functional studies. Functional studies, although the requirement for such change from differentiated cell types to another is not likely to occur in normal development, such phenomena known as transdifferentiation has been achieved experimentally in Amphibia. So in this case, we can see that there is some tissues or some cells can be can change their function from one function to another function. So this called transdifferentiation. So we see, before we couldn't, scientists couldn't understand that how a cell, differentiated cell and specialized cell to transform its function from one function to another function. So this have been observed actually and proved experimentally in many organisms, including plant and amphibia. We will see, for example, in plant, such regeneration, if you study tissue culture, for example, you can see this regeneration, and during the regeneration process achieved in several species, including both in carrots and tobacco and actually many other plants. For example, you can see in carrot that you can take some tissues from the phylum and putting with some nutrient in tissue culture, and then this can be actually transported in the plant. So you can see, for example, here in the example plant, you can see that if you take some tissues from a plant, from mature plant, you can take from the phloem, and then you can take these cells and take these tissues and just plant these tissues on the right media, in the tissue culture media, and just put some hormones and that produce like auxins and cytokinins and all that, and just put these hormones on the media. Actually, this somatic cells transformed into a germ, something like a germ cell, and these somatic cells can produce a plant that is differentiated in different tissue. So this we observe this in many plants, in many kind of cells, from different kind of cells in plants, and many plants have been used to produce other plants by this phenomena, which we call it totipotency cells. So this is the totipotency cells means that that cell is capable to produce a full plant from one single plant. This phenomena also have been observed in Amphibia, and we can see here that if you just take a nucleus from a specialized cell to an unfertilized egg, and you removed the nucleus by UV, simply this nucleus from the differentiated cell can be located here and then give another copy of that organism. Actually, this is the idea of cloning, and have been used after that in 1997 by scientists to produce Dolly the sheep. So actually that means that you can take the whole nucleus which contain all the genetic information from one organism and put it in another egg that have no nucleus, or other cell that have no nucleus, for example, and then put it in the right condition and in the right place, and then you can produce a full organism out of that by different development, by normal development. So this is the second type of study, which is the function. So now we'll talk about the third level of study, which is molecular studies. In this case, we use probes, can be used, gene probes. It's, for example, Southern blotting experiment to investigate the structure of particular gene in DNA prepared from different tissues. As with the use of northern blotting, West and Western blotting to study respectively the RNA and protein, Southern blotting can be used to investigate the structure of DNA in individual tissue. So now we'll see that this kind of molecular techniques, which we'll talk later about the difference between these different techniques, are used to prove that there is no loss of genes are observed in specific tissue. So in Southern blotting, actually what we do is digest the DNA with restriction endonucleases, which cut it into a specific sequences, and then we make electrophoresis on agarose gel, and then we transfer the DNA in the gel to a nitrocellulose filter, hybridized filter with radiolabeled probe for gene of interest, and then analyze structure of specific gene based on pattern of hybridization obtained when DNA is cut with different restriction in the nucleus. So it's very simple technique, and this simple technique just prove for us if there is loss or no loss of particular gene are observed in a specific tissue. So we have these actually to prove if this exists or not exists. In this case, we need to just see the principal differences between southern, western, northern, and dot plot techniques. If we take a look at the comparison between these three techniques, the southern, with northern and western analysis of gene X, we know that the gene actually transcribed to mRNA, and mRNA is translated to a polyprotein. So simply, if we know that the DNA, if we prepare the DNA sample and we make a radioactive probe, for example, or labeled probe from DNA segment, we can simply detect if this DNA exists in this cell type or not. For example, we can extract the DNA from a particular cell type and then after electrophoresis and separate them in the gel, we can just transfer the segment onto a nitrocellulose membrane, and then we can put the probe, the gene probe, and see if the gene probe hybridize to the cellulose membrane. This gives us the evidence that this DNA segment exists in the cell, this cell type. From the southern, we simply in the northern we talk about RNA. So we take all the transcribed RNA in specific tissue, and then will be put in electrophoresis, and then this from electrophoresis transfer to a nylon membrane, and then from the membrane we can see if it hybridizes with a labeled mRNA or labeled gene that we need to see if it exists or not. The same thing with Western, but actually in the Western we talk about isolating the proteins of the cell type. So simply that is, as we said before, in the Southern blotting, such technique was developed. The name of the scientist is Southern, and this E.M. Southern, that this technique have been developed in 1975, and the Southern technique, as we said, we can see that the blot is used to detect presence of particular DNA fragment. This is number one, and the principle is hybridization process of forming a double-stranded DNA molecule between a single-stranded DNA molecule probe and a single-stranded target DNA. There are two important features of hybridization. Number one is the reaction are specific. The cell probes will only bind to target with a complementary sequence. Number two is the probe can find one molecule target in a mixture of millions of related but non-complementary molecules. So this, by Southern, we can simply detect that any existence of any DNA sequence in specific tissue type or cell type. Northern blotting, actually, this technique is for detection of specific RNA separated by electrophoresis by hybridization to a labeled DNA probe. It's Alwyn and his colleagues developed that technique in 1979, and also here we can see that the principle also is hybridization. So, but here with the working material here is RNA, not the DNA like the Southern. And we call it Northern because actually the DNA technique we call it Southern. So that's why when they try to make a name for the RNA technique, detection of RNA, so they call it Northern. The Western also, this Western is working actually by detecting specific proteins separated by electrophoresis, so that by use of labeled antibodies. In this case, this technique have been developed by Tupin in 1979. Principle is antigen-antibody interaction. So simply we extract the total protein content from a cell and then separate that total protein content of the cell on gel electrophoresis, also protein gel electrophoresis SDS page, and then after making this SDS page, simply we can add the probe to detect if this protein exists or not. Now to the technique number 4. This is called dot plot. Dot plot is simply we can use a non-fractionated or non-electrophoresed sample are directly plotted on and immobilized on nitrocellulose or nylon membrane as dots or spots for hybridization. So we can take sample from different sources, can be tested in a single run Also, the principle of that technique is hybridization. So we, for example, we can take from sample number 1, number 2, number 3, and we have the same probe and see if that probe exists, how much it exists in each of that cell types. For example, if there is a dark spot, so we can know that here the probe actually much more hybridized much more than that first one. So we can see that there is differences in the density of the spot or the dot, which means that there is much more intensity existence of more existence of that gene, for example, or that whatever here more than here. So this is a technique called non-dot plot, and as we said, it's non-fractionated or erythrocyte sample. So now we come to the practical. We need the student to describe cases of loss of nucleus in mammalian cell during differentiation and loss of chromosomes in mammalian cell. That the example that we just presented to you. And in question number 3, we need to give us example of the transdifferentiation implant. Give us example with a drawing. You can give example what the main describe the cases which you can see transdifferentiation implant. And the other question, we need the student to describe the principal differences between Western blotting, Southern blotting, Northern blotting, and dot plot. What are the major differences between these four type of techniques? Subscribe to the channel and have a good time. Our video, other educational material. Thank you and see you in the next lecture, inshallah.